Okay. 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 11:15 o'clock. We start on time. Good morning. My name is Witold Walke. I have the honor to lead a session, Biomaterials of the Future. There will be 4 presentations during this session. I'm convinced that this will be very interesting issue in the field of biomaterials engineering, biomedical engineering, and the first speaker. I invite the first speaker, Professor Andrea Sorrentino from Italy. You're welcome.
Thank you, Mr. Chairman. Thanks to the organizer for the opportunity to present here some results of my current research in this topic. Let me start this presentation. No. Okay. Let me start this presentation from here. I want to start from this inspirational sentences. There is no. Okay. Okay. These inspirational sentences that I found outside of this room, in which well describe the activity that we make in our research center in Italy, in the north of Italy, Lecco, where we are in the same building, 7 different institutes that work in different topics and have different research interests, but all together, time to time, we make a project with a very specific objective, and the activity was shared between this institute, and with this, in this way, we are able to reach from the laboratory up to the commercial application of these results. For that reason, we will see here that I show you some results in which I am not directly knowledge, but it was possible for me to do that just because we share this research all together. Of course, it was also possible because we have a strong connection with the territory of Milan territory, in which there are several hospital and clinic centers that are more involved in these fields. Let me start with this first research, in which we dedicate our time to apply to the scoliosis. It affects about 1 million people around the world, so it's very, very important pathology that affects especially young women in a very precise moment of their life, that when they want to construct connection with other people. So it's important for these people to have all the possibility to share their experience with this, and have this braces, that is the only therapy that at our moment is known and work with this type of illness. It becomes very complicated. Image that we have on your body for 22 hours per day, these shields that have no transpiration, is heavy, and have no very strong aesthetical effects. What is the reason for that? But there are some problem in my mind. There is some problem related to the factory that make this type of object. They generally are a small factory that work close to the clinics. They generally work with a very small amount of items for annually, generally 1 or 2 items for days. So for this type of situation, it's very complicated to improve in equipment, very expensive. So they prefer to use manually approach, in which they start from the negative of the patent, just around the patent, by gyps capture. Then they make the positive, and again with thermoforming of a plastic sheet on this positive molds, they obtain the results. After that, you have a lot of work for finishing this object. Of course, they have to use a plastic sheet with constant thickness, and have no way to work on the aesthetic of this. Fortunately, in the last years, due to the reduction in cost on the laser scan, some of this part was changed. Now it's quite common to find scanner, body scanner that is able to scan the body of the patient and give you the at very high resolution the surface of the body. After that, there are already on the market some software that is able to match together the surface of the body with the X-ray of the skin, so also algorithms that optimize the design of this object is possible to obtain a finite object without any particular competencies. After that, they give this file to a CNC apparatus that work on a big cylinder in polyurethane and make the positive molds of the patient. After that, they make the usual thermoforming approach. It's clear that some of the problem related with the waste generated with the gypsies was in some way avoided, but there is waste related to thermopolyurethane working, and also there are problems already present for the manufacturing of this thermoforming sheet. It's clear here, I image that all people that works with the last advantages is in 3D printing, thinking, why don't use 3D printing for this type of application? Well, the reason is that we already have several problems with this type of technology. Problems are related with the inconstant quality of this product and with the dimension of the object that we are to create. Also with the materials actually available on the market that already present some specific problems. But probably the most important aspect related with this process is the fact that you can change point to point the geometry, the thickness, the structure of the object. You can reduce a lot of the weight of the object, and you can insert also additional feature in this object. But what is the problem that I mentioned before is related to the anisotropy of the material that you obtain when you use this type of technology. Anisotropy is common to every type of additive manufacturing process, but in the case of DM is more and more important. Just because image you are put one on another, two layers of material that are at different temperature and in different condition, and you have to hope that these two layers have to stick to have a good addition between them. It can be improved, of course, by increasing temperature or change some parameters, but generally the difference is not so high. If we try to compare results obtained with the classical compression molding and 3D printing, you found that the differences in the mechanical properties are very important, especially in the zeta direction, the direction of growth of this part. Of course, you can change the position of your part in the printer. You can you can have so lucky to align the load direction of your object with the perpendicularly to the growth direction. But also in this case, it's very difficult to obtain results that are comparable to that we found with other technology. I don't know in which way I can practice. This is a video, but now works. Okay. This is a video in which I show the use of self-healing materials. This material is able to recover completely their structure, autonomous or by external stimuli. In particular, I'm a researcher in material science. We develop composites in which we use, for example, polyethylene co-methyl acrylic acid. This is a small molecular weight filler that we use in our composites. It's able to self-healing the structure at very low temperature that it's more or less the same of the body weight or the body. So it means that the patients during the use of those have a continuous rejuvenation of the object that they have on site. And of course, also during the printing, the presence of small molecular weight have the ability to diffuse around the layer and increase the addition between the different layers. See, allow to completely recovering of the mechanical properties of this system. Okay, this is just one example. Let me show you what is the effect of the anisotropy in this type of material. If you imagine to print a conductive material, like a material that you can obtain by thermoplastic, common thermoplastic material in which you are putting filler in the nanometric shape, then you reach the right composition that is higher than the percolation threshold, you obtain conductive materials. Okay, if you print this and you change nothing, just the direction of printing, you obtain completely different values of the electrical conductivity. So you can go from a conductive materials to an isolant. Okay, of course, conductive, don't think to the metal conductivity. And when I speak with the isolant, is not ceramic, of course. But for us, it's enough for our application. You obtain very, very different effects, and are on the order of 2 orders of magnitude or this effect. Well, it's not a problem in some cases. For example, you can use this effect to obtain smart materials. For example, you can use heat for making self-sensing materials. In particular, when you apply a stress on this type of material, the stress is able to remove or changes the composition of the material or the alignment of this filler inside the material. So you can change the electrical properties of these things. So following the electrical conductivity of this material during a mechanical test or during a normal use, you can obtain a continuous monitoring on the property, on the condition on the materials. So with important information what happens to your items. Also, you can use it for thermoregulating the systems. If you apply a voltage on these materials, just because there is conductivity, but it's not conductivity, I said before, as a metallic, there is a Joule effect. So you have read an increase in temperature. Let me show you some example. Sorry, but I'm not able to start the video. Okay. Okay, thank you. As you can see here, the video, this is a very, very, very simple. Okay, there is a the bar that's not. Okay, this is a very simple sample in which was obtained with the same material, but changing just the direction of printing. In an half of the pieces, there is a direction of printing. In the other part, there is a orthogonal direction. So it means by applying the voltage, you obtain an increase in temperature in just one side of your sample. Of course, during time, the heat diffuse for thermal effect, but it's not due to the generation of the heating that apply just in one side. It means that if you are able to design your object, making the right direction of your printing in the different zone, you can manipulate or you can regulate the temperature inside of your object, just doing this and using the same material for all the object. As you said before, also you can follow the tensional inside your sample by changing or by following the electrical conductivity of your sample. Just very simple experiment in which mechanical properties you can follow the deformation of your sample from a mechanical point of view and the change in the electrical properties on your sample. It's follow more or less quite well up to the elastic deformation. After that, there is some plastic deformation inside that are not recovered. But time to time, this deformation it's enough for our scope. Okay. So by change materials, you can apply different functionality and you can improve your results. These, for example, are results that we are obtained with a patient, with a clinical trial. We demonstrate that it's possible by 3D printing to obtain objects that can be really used on patients. it is very, very simple geometry that just for start. What we see that with this material that have a particular future, that not only the material, but also we cover the material with the particular substances that avoid, for example, the formation of smell, because during the use, there is an increase in humidity inside. There is a formation of biofilm that start to smell after some days, and also for thermoregulating the temperature on the body. You can change the comparison between the commercial braces and that we obtain by 3D printing is completely different. Okay. In the moment that we demonstrate that it's possible to work on it, it's possible to start to change the shape of this object, trying to improve the aesthetic aspect of this and improve the wearability. But also we can start to thinking on different structure, on different materials that work together to give different opportunity. And also, for example, for thermoregulating, you can work on the structure of the object by using the 3D printing for making void inside shell core and introduce thermoregulating materials, and so on and so on and so on. Just to image what you can do with this. And lastly, it's important, or another possibility is to follow the patient by telemedicine. It's possible to improve, to fix on the structure sensors that are able to communicate for ever a continuous evolution of the patient, the use of this that you can follow. You can see if really the patients follow the therapy that you have written, and again you can work. There is several examples. This is another example, another application. It's not have the same results of the previous, just because it's a smaller one, and people don't think that the application 3D printing in this case can have a good effect, just because they prefer to have a rapid approach with the traditional approach in that case. So it's also important to think on this. Not only the solution are well in the case. This is another project in which we want to demonstrate that it's possible by 3D printing obtain object device that can prosthetic foot that are used for normally life for people that have a high level of activity. It means young people. In this field, the level of activity is K1 is elderly, K4 is the maximum activity or athletic activity. We are here in the fields of K4, and for people that have a weight in the order of 80, 100 kilograms, so very important performance for this. We want to demonstrate that by 3D printing it's possible to do that and to obtain the same performances in terms of biomechanical behavior of this object. To do that, we have to use continuous fiber, continuous filament fiber 3D printer. It's a quite new technology. It's formally similar to what generally was used in some industry, automatic fiber placement, but in that case we use thermoplastic material, not thermo settings, and we don't use molds, and we cannot apply pressure. So for that reason, we have a strong problem related to the presence of void inside the structure that we are printing. In any case, knowing all the aspects, another important aspect is that at the moment just 2 companies around the world have this type of technology due to the patents. So we try both technology, waiting for, I think in a few years, the patent was died, and then we start to perform with our apparatus with the results that already we have obtained. As you see, the material that they use especially this technology, you have to use the material that they give you. You don't have possibility to have another type of PCB or materials. And the quality, this is a cross-section of the filament. In that case, this is the carbon fiber, this is the polymer matrix around. There is a very low level of carbon fiber inside this material, and the quantity of polymer inside is not homogeneous distributed in the filament, and of course the results in terms of homogeneous when you print is not so good. In that case, the situation is better, but more or less the problem remains. In fact, when you try to make a fatigue test, that are the tests that can give you the information about the durability of your material in a real situation, you observe that your samples collapse. There is no collapse of the carbon fiber, but between the shear, between the different layer of materials. So this have a strong effect on the durability of your samples. And this is a microtomography of the sample. You see there is a lot of void inside, about 50% of void inside. In any case, also changing the printing condition and applies. The only way that we found to increase the properties of this material is to use a post-processing by hot compactation at high temperature. So after printing, we have to put under pressure our systems to increase the density of the material, reduce the number of void inside. In any case, knowing all this limitation, we have developed a continuous system in which you start from your geometry, global geometry, then the system is able to have a partial section of this geometry. You have inside to have the structure with the position of the continuous filament of fiber, and then the simulation of the structure in order to have the results in terms of mechanical resistance. This was a loop. You have continuous to do that in order to obtain the optimization of your final geometry. This is some evolution of what we are thinking at the beginning of the project. We are doing now, more or less. There are other results. Of course, all these projects have to have in mind what are the limitations of the technology. In particular, the position, the bonding plate, and the number of filament or the dimension of the filament, and a problem like this. But this is just one aspect. Another aspect is related to the biomechanical properties. You have to simulate, or you obtain the similar results that you have with the commercial apparatus. So, for example, in that case, you have to mimic the rollover shape. The rollover shape is, it's difficult to explain, but it's a measure of the evolution of the work of the processes during the working. If you follow exactly the rollover shape of your foot, you can have a movement that is equilibrated without static or step during the movement. That it's important if you want to ensure that the patient have a normal life, a normal behavior during their working. Our test was also making on clinical trial that allowed to obtain a lot of information regarding the pressure on the healthy limbs and on the prosthetics. These are very complex shape. I'm not an expert on gait analysis, but you can obtain a lot of information about it. And what I know when I see this is that this curve have to be closer as possible. Just this means that we are mimic in some way the behavior of the foot and the commercial prosthetics. Of course, gait analysis give you really an incredible number of data, of information, and my colleagues are trying to use all this data that come from different sources from pressure platform, for optical system, for sensor on board on the device, for making analysis of the data with intelligent machine learning, obtain a complete picture of your patient and the effect on the patient or the change that we make on the device. It will help in the future, for example, to follow the evolution of our systems, and also have an idea when it's time to change your device, or it change to go to the clinic for changing somehow, for making some fixture on the systems. Just because you use these every day, so the changes are small and continuous during time. You don't, the patient have no chance to have an idea what happens really. So the only way is a continuous monitoring of what happens during the normal use of this. As you see, there is a lot of information. Well, also in this case, it's important, it's really important to make fatigue tests on this apparatus. The number of tests have to mimic the use. Our objective is to reach at least 3 hours, 3 years of continuous use. At the moment, we are in the more or less 1 hour, 1 years. It's not enough, but the price of this object is very low with respect to the commercial one. It means that you can change it more quickly, and also the fact that we use thermoplastic materials gives us the possibility to recycling this type of object and reorganize it for a longer life. Also, the results obtained in terms of failure give us a lot of information regarding where it's important to have some changes for improve the quality of our object. Okay, this is the evolution of the project. Now we are trying to integration of sensors, other objective on the system, and try to use a customer system that can follow the pattern and give a complete picture of the evolution, the life of the pattern with this. Okay, this is another example in which I'm not directly involved. This is the development of a fixture for tombs amputation. This is a very special case. The number of people that have need of this type of therapy are very small. In Italy, they tell me that more or less there are 100 of people per year that have need of this type of application. For that reason, there is no specific object for this type of problem. The people that want to use it have to use a material that come from odontoiatric application. So you can understand that you don't have the right dimension of this object, and there are also problems of compatibility with the system. Okay, my colleague that work with the metals make a lot of tests by changing the direction of grow, changing the materials, changing the structure. For example, I remember at the beginning they start with a very complex geometry inside with lattice geometry and Voronoi and so on. But at the end, what we found that its best result was a standard geometry close. And what is important is the surface of this object. Is necessary to have the right dimension of the roughness on the surface in order to avoid to allow the growth of the cellular on the surface. Also, here you see, for example, all the particles that are not attached on the surface must be removed. It was very difficult, this step. Also, we have to ask to company outside of the project for cleaning this by sun, because they have the right equipment and the certification that ensure that no free particles are present on this object. Image the dimension of this particle is very difficult to manage or to use. In this project, our TV require the formation of a biofilm on the surface on this object in order to improve the compatibility of this fixture and allow or improve the growth of the cellular, also with using antibacterial component. Okay, after this coating, we make several tests in order to prove that it works. So tests that are special designed for clarify the effect of the coating, the resistance of the fixture, and the normal use during the practical. This is another project, very at the beginning of this project. We are trying to develop a social bike. This is a bike that have to stay connected with virtual reality. So for elderly people that to stay at home and have no possibility to go outside, they can go together with other people, with other friends that are present in other place in their home. They share the experience to go around. Also, there are some tests that have today in order to improve the exercise, physical exercise. In our case, in that case, we have to develop the saddler with a new geometry in order to improve the use of this by elderly and allow the use of sensors on this in order to have a continuous monitoring of the position of the patients on this. Okay, let me just 1 minute more to show you the conference that we are organizing the next year in Salerno. I hope that you have 2 topics specially devoted to the biomaterials and device for biomedical application. I hope that you enjoy with us, and thank you very much for your attention.
Thank you for your presentation. Thank you for very interesting lecture. I'm opening the discussion. Yes.
No, I don't need that. I've got it really thank you. First of all, congratulations, and I'm really impressed with your presentation. And I have a question related to braces you mentioned. I mean, that there are different parameters that can be applied, as you mentioned, say, some thermosetting, etc., etc. And have you performed tests, let's say, somehow quantifying which factor impacts the outcome most? I mean, is it like, for example, the different material? Is it just a shape? Is it like thermodynamics, or what could be?
Okay, we try several type of material. You have in mind that also this is a medical device, so you cannot use every type of material. You have to use just few type of material. In particular, you just can use PLA, but it's not really okay. You can use PLA, you can use PET, polypropylene, and I think not more material that are normally used in 3D printing. So you cannot have a very large number of material that you can use. Of course, by changing, for example, the temperature or changing other parameter, it's possible to have some improvement in the mechanical properties, but we have to in mind, when we start our project, the first time when I print the device, it take, if I remember, 100 hours longer. It's impossible to have so longer time. At the end of the project, we was able to print in 20 hours. That it's a very long time, but it's still compatible with the workflow of this type of clinics. The patients that reach in this factory generally stay for 2 days. The first day they take scanner on the body, and the second day they have to prove the brace, make the changes in the last fixture, and then go home. and then you cannot have more time to prepare your praise. So if you make the price, if you make the time necessary to print, if you make all these together, you have very small changes to change for obtaining better results.
Thank you very much. Unfortunately, not for the research, however, for generally speaking, unfortunately, you touched the point, the money, which is for sure the factor for us for research. You know, it shouldn't be taken into care. However, since we are operating in the living world, so for sure the price for the final medical device plays a crucial role in some examples. Thank you so much.
Thank you. Okay, I have one question. What kind of material, in your opinion, in the future will be better, will be perfect for prosthetic devices? Maybe metamaterials, maybe material with a structure oxetic, maybe another, I think.
On the device that you are referring. One of our objects in LICO was to have the patient at the center of the object. Then we try not to have the best therapy in absolute. We are trying to make the right therapy or the right device for the specific patient.
Mm-hmm.
So one of our objectives is try to reduce the price.
Yes, okay.
So we try to use a simple process material that are simple, that can be used and reused. So the terms maybe is the sustainability. Of course, in some special cases, when you want to reach some high level of performances, especially for example for prosthetic foot, you have to use a special material that can give you some particular. Now I'm thinking on one work page or this project in which some colleagues are working on some metamaterials, memory shape alloy material, combined with other material that in order to improve the energy storage. That it's really important, especially if you want to use this for longer. It's an opportunity, but very expensive, very difficult to make.
Yes, understand. Thank you so much. Thank you. Okay. The next presentation will be presented Agnieszka Antończyk from Silesian University of Technology. Microphone is yours.
Mogę do mikrofonu, czy... Okay. Good morning, ladies and gentlemen. Today I would like to show my research. I'm a researcher in the Silesian University of Technology in Faculty of Biomedical Engineering, exactly in the Department of the Biomedical and Mechanical Devices. Today I would like to show my research, exact application of spherical aluminosilicates as the fillers in the composites for medical applications. First of all, I would like to talk about microspheres. Microspheres is a thin, small particles with different kind of sizes, start to a few micrometers up to 100 millimeters. It can appear solid or the hollow. Nowadays, probably microspheres appear in every materials in our life. For example, our toothpaste to concrete institution. But we have a different type of microspheres. First of all, it can be polymers. It's the common. It can be PMMA, PLA, and different kind of biocompatible used exactly in cosmetics industry. Another, it can be ceramics, glass microspheres. Here it's especially thermal insulation and aerospace materials. Another, it's metallic microspheres. in this type is also a carbon microspheres, or can be covered by different kind of carbon or nanoparticles. It can be sensors, catalysis, and other composite microspheres. It's combination with different kind of materials. Spherical aluminosilicates. It's just more natural microspheres because it's this history. It starts in hard core. And then next step, it's the carbon. It's from the hard core. It's combustion in power plants. During the high temperature and the forces during the combustion, it creates a hollow microspheres. It's look like this. Rest of the flash and the rest of the combustion, it's throwing to the lagoons or the water reservoir. What's going up to the surface, it's exactly the spherical aluminosilicates. This is a hollow microsphere, so that it means it's ultra light microsphere, but have a high resistance to chemical agent and high and low temperatures. Also have a significant compressive strength and resistance to crashing, and also have the ability to damp vibration. Why is it is very important? Because this can use to filler in the concrete nowadays, and also filler in the paint and coatings and different kind of industry like abrasive materials. But today we talk about biomedical engineering. That kind of filler can use different kinds of materials, start to scaffolds for tissue engineering, then the biocompatible composites and fillers, coating of the implants or creating implants when we talk about combination with metallic matrix, and also, also drug delivery. But if we want to mix it with some kind of matrix, for example, the polymer matrix, because it's so much easier than the metallic matrix, it is very difficult, because that kind of material, that shape and the surface, it's very glad. So we must change the surface to have a good bond with the matrix. How we can do it? We can do it with the silanization process. Why? First of all, the surface modification, it's by bonding with the nitrogen. Next, surface modification by increasing the number of carboxyl, amine, and nitrogen bonds, and increasing mechanical and chemical bonding for composite application. In here, it's an example of the silanization process using organic sealant. It means 3-aminopropylotritic sealant, means APTES. But nowadays, I want to show you my research using silicon nitride with sodium hydroxide. But the first step, we must changing the surface using a very aggressive acid. It means caro acid. In here we have changing the first step of the morphology, and after this is the process of the silanization when we bonding chemical groups. That I say, I use it silicon nitride with sodium hydroxide. And this is the final version of our microsphere coating the silicon nitride. I say I want to create new type of composition with polymer matrix, so I choose bond cement. Why bond cement? Because currently used bond cement exhibit low mechanical properties, then have a high matrix polymerization temperature, can lead to bond necrosis, and also have toxic monomer can lead to a wide range of adverse effects of the cardiovascular system and the rubber. So, the create bone cement based on the spherical aluminosilicates can approximation of the mechanical properties of the cement to the biomechanical condition of the skeletal system. It could reduce the proportion of the matrix, including residual monomer, and improving biocompatibility with the bone. So, in my research, I choose PMMA matrix and have a 3 variant of the combination with 20, 30, and 40% amount of spherical aluminosilicates with the modification. This is how it looks like. A, it's PMMA without the aluminosilicate. B, C, D, it's 20, 30, and 40% amount of microspheres. I also combine it with hydroxyapatite. The first, it's without aluminosilicates. This is still PMMA. And the second version is with 30% of aluminosilicates. We see the changing of the structure and we see some visible of the small particles. I also check with using microscopes, scanning microscopy, to see how the morphology is changing and how much silicon is appear on the surface. First, it's without filler. This is only PMMA. In here we have see the particles of PMMA. In here we have a 20% amount of aluminosilicates. In this we have silicon appear. This is after modification. We see this all the surface, and this is amount of 30%. In here we have some agglomeration of our aluminosilicates, our filler. It's also appear with 30%. Of course, it's appear more. So they conclude the best amount of the filler it should be to 30%. I also check the polymerization temperature because this is very important when we talk about the necrosis of the bone. So in here have a very funny situation. Why? Because I repeated 3 times, and every experiment I have a different results. So now we can say definitely what kind of filler it's decrease or increase the temperature, but every of the experiments say that around 5 to 7 minutes during the polymerization, the temperature going up to 80 Celsius, but during exactly 1 minute, it's during 30 seconds, the temperature rapidly rises to 80 or more degrees, and at the same time after 50 more, maybe 20 seconds, rapidly down. So that it's very short time that the temperature is very high. But in the newest research, say during the endoprothesis surgeon, when the bone cement it's during the polymerization, the surface of the implant is heating. This is a good common because it can be destroy the biofilm and help to amputate therapy to the patient. And I also check the biological tests, the cytotoxicity. I use LDH experiments. So after 38 hours, the amount of 13-40%, it's the excellent version of the next experiments. To conclude, the use of aggressive acid allows to change the morphology of the surface of the samples, which allow to increase the surface interacting. The results showed the feasibility of creating a mechanical and chemical bond to the composite matrix, and also using spherical aluminosilicates in the composite reflect the polymerization curve, which shows invariability in the polymer's cross-links process. And the study shows that bond cement in vitro tests showed that with 30 and 40% that I say, spherical aluminosilicates was the most favorable option compared to pure PMMA. So that amount of aluminosilicates, it's be better than to use it. So thank you very much.
Thank you. Would anyone questions? No?
Thank you.
Maybe during lunch. Okay.
Of course.
Thank you.
Może tu zostać.
Okay. I invite to next presentation. Speaker is
Good morning, everyone. My name is Aneta Dyner. I represent the company Chirmed and Cheerstone, and I work in these two companies as a sales and certification director. So today I want to present the results of FIER's project Atrauma Biomat. Advanced metamaterials dedicated for cardiovascular surgeries to minimize tissue injury. So it's a 3 years project. We implement this project within an international consortium. So we are having nine partners within the consortium. So I am very sorry, but today I will not be able to answer all the questions because it's a very huge project with a huge number of results. Maybe some question I will be able to answer, because I don't own the original results of the studies. So we within the consortium we are having Silesian University Faculty of Biomedical Engineering, the company Cheerstone manufactures surgical instruments, who is the industrial partner of the consortium. Professor Zbigniew Religa Foundation of Cardiac Surgery Development, Institute of Metallurgy and Material Science of Polish Academy of Sciences, Jan Długosz University from Częstochowa, Joanneum Research from Austria, Kocaeli University from Turkey, PCCL company and Distech company from Austria. So this is the graphical abstract of the project. So the goal of the project is toolbox technology for a wide range of tissue non-injurious cardiovascular and thoracic clamping and grasping instruments. So we are working on atraumatic instruments. So here we want to integrate the idea of metamaterials and sensors for tissue overload protection. And we are also working on super hydrophobic, easy to clean surfaces, which is very important for reusable surgical instruments. And our goal is to work on sustainability, so the design for reuse and design for repair. We are also working on full biocompatibility with all solved critical issues for medical device approval. And also within this project, we are approaching the certification. So we want to take this challenge to prepare for the certification, because in this time where we have the very strict certification rules, we want to work on it within the project to work also on the commercialization of the project results. So the main problem in the existing instruments are uneven clamping forces on the whole surface of the clamping parts of the instrument. So this problem has been solved already for big instruments, like you can see here. So here we can see the Kowalski clamp. So this solution was used only for bigger clamping instruments. So there are still some solutions missing for small micro clamps and also for laparoscopic instruments. So the main issue for the clamping instruments. For the vessel clamping instruments is to close the vessel with the minimum required force. So the force which is required for occlusion of the blood vessels. So therefore we performed numerical simulation and also performed experimental verification of the results to define the minimum clamping forces just to close the vessel. And the main findings, so we defined these forces, and the main clamping force largely depends on the vessel internal pressure and also diameter. We also performed histopathological evaluation just to show that if we use the excessive clamping forces, which are higher than the defined occlusion forces, we are getting major tissue damage. Here you can see native tissue not stressed and also tissue after the pressure sampling with the application of the excessive clamping forces. We also performed numerical simulation for the clamp design. So these are some examples, and we also did the demonstrator of the instruments. We used two technologies. We used 3D printing, so PBF technology, and we also used traditional CNC machining. So we got better results for the traditional CNC machining. We got a better characteristic of the surface for this clamp. So this is the picture of the first demonstrator. So this is the principle of action for laparoscopic instruments. So also we are relying on the idea that the clamping forces, they should be parallel on the whole working surface of the instrument. So within this project, we are working on reduced tissue traumatization. So we also developed an atraumatic insert. So the atraumatic insert consists of two layers. So the lower layer is built of titanium alloy. So actually, also the whole instrument is built of titanium alloy. And the upper layer is built of elastomeric material. The elastomeric resin was used for the 3D printing in the technology of the low force stereolithography. So we used commercially available materials, Formlabs Biomet 50A and Formlabs Biomet 80A, so with two different shore hardnesses. And as you can see, the insert has also the serration. So we also developed the serration on the surface of the inserts just to avoid slipping the instruments from the vessel. We also investigated biological properties of the material biolabs. So I'm not showing here all the results because there is a huge number of the results. Here is only the summary. So here you can see that we got very good, promising results. So it proves that we can use this materials for reusable surgical instruments. So we tested cytotoxicity on LDH in direct and indirect contact. We also considered two different ways of sterilization, so low temperature and autoclaving. We also tested pro-inflammatory molecules, and we also did microbiological testing. So we confirmed that there was no germ growth on the tested surfaces, and we also performed a degradation study in simulated body fluids. So within this project, we want to work on the materials which have the ability to adapt to the excessive clamping forces, and therefore we decided to use metamaterials, which are architected materials that derive unique mechanical properties from the artificially designed internal structure rather than just their constituent materials. Here is only the example of the metamaterials. This is not the material we used for the study. We just wanted to use the idea of pressure matching mechanical metamaterials. We performed a large number of numerical simulations, so for the inserts both for laparoscopic and traditional instruments. And we chose several different geometries both for laparoscopic instruments and traditional instruments. So it may look very strange because when we are thinking about the metamaterials, so we think about the lattice metamaterials. So here we have some limitations because the inserts are really very small. So we cannot build huge lattice structures. We also performed insert simulations. So in almost all simulated inserts, we got the stress plateau. So it means that the stress in the artery is independent of the applied force. So this is what was very important in terms of the project goals. We did a microscopical evaluation and also histological evaluation. So for the 3D printing process, there are still some issues. We are working to optimize them. So the printing accuracy is not as we wished. So we are now working to optimize. We also, as for the histological analysis, so despite of stress absorption, so tissue damage was not eliminated. So here we are seeing reduced tissue damage from the level mild to moderate. However, we have the ambition to eliminate the tissue damage completely. So now we are working on the new geometry of the metamaterials, just to improve the overload protection of the tissues, and we also want to optimize the 3D printing setups. Within the project, we are also developing hydrophobic coatings to make the cleaning of the instrument easier. We developed 4 different coatings. They are consisting of HMDSO and PFOTES in several different configurations. So we have 100% HMDSO, and we have also configuration 90 to 10%, 50 to 50%, and 10 to 90%. So we also performed a huge number of the studies for these coatings, but these are the main findings. So we observed the wrinkling effect, so increasing the proportion of PFOTES in the mixture of HMDSO leads to increased surface roughness and waviness. We can also observe wear progression starting from the combination 50 to 50 onwards. And we achieved the hydrophobicity versus initial states. So we are still working to get the super hydrophobicity in this case, but now I think it's quite a good result. So the main finding is that the combination of HMDSO and PFOTS allows for obtaining a thin coating with optimized adhesion, corrosion resistance, hydrophobicity, and bioneutrality, which makes it particularly attractive for surface modification on medical tools and implants. Within this project, we have also the ambition to develop sensors. What's the purpose of this sensor? So we want to have a final protection against excessive forces. Here we want to use the PiezoFlex technology, which is a patented technology invented by our partner from Austria, the company Joanneum Research. Here we can print. The main idea of this sensor is that we can build it as a coating on each surface we want, and also so we have PiezoFlex can work in as piezoelectric sensor, and as a piezoelectric sensor it can detect very small changes of pressure. We also did the degradation study. We included the conditions of reprocessing for surgical instruments like cleaning and sterilization in autoclave. So sensors are very resistant to high temperature. So even in the temperature 70 Celsius, so no visual changes of the sensors were negligible. And also here we can see the sensors after AR spectrum after 2 months incubation is 70 degrees, and also after cleaning and autoclaving. And here we can observe that we can maintain chemical integrity of the structure for the study period. And also cleaning processes, repeated steam sterilization did not affect the structure of chemical bonds within the samples. So as a further research, we plan to develop a sensor which are adapted to the tool dimension. It's a real challenge because the micro clamps, they have a really thin surface, and we want to develop the whole demonstrator. So a tool plus atraumatic sensors and coatings. And we also do the final demonstrator assessment in vivo-like condition simulation. And of course, we want to prepare the whole idea for certification. So thank you very much for your attention.
Thank you for your presentation.
Thank you.
I'm opening the discussion.
Thank you. And first of all, congratulations for presenting this very complex issue in such a comprehensive way. So thank you very much. It was a really pleasure to hear that. However, I've got a question regarding coatings. You mentioned that these coatings were used as a perfect solution, finding a solution in optimization process to assure proper automatization. However, due to, I was wondering if you checked deformability of these coatings.
No, not yet. It's under evaluation.
Okay,
yeah.
So it will be tested because it's very important and it's very interesting about, the behavior of that one. But if it hasn't been done yet, but you've got it in the plans, so this is very.
I'm also very excited to get the next results because I haven't found this kind of coatings for surgical instruments. So I'm really curious about the next results. Thank you.
Thank you. I have one question.
Yes.
Very interesting material. Do you think it could also be used for implants in future or not?
So it depends which idea you mean, because there are several ideas within the project. So you mean the coating, yes? Mm-hmm. So at this moment, I cannot say because we don't have so many results. So at this moment, we are focusing on getting the super hydrophobicity for easier cleaning. So at this moment, we cannot say whether it can be used for implants, but I think this kind of coatings are very promising because, as you can see, we can do the combination of the ingredients here. So maybe we can also optimize the coating for implants and also the good. So they are also good in terms of biocompatibility. So they are biologically neutral. So I think they are promising also for implants.
Okay, thank you.
Okay, so I have a question about the sensors because maybe I don't understand. Because you say that the sensor is to know how much stress you have, and how do you know what the stress will be? Do you have a screen? How the sensor will working to measure the stress?
So we want to. So actually, I didn't show the whole system. We want to build here, but he will of course need a kind of. We want to have like a Wi-Fi connection with some electronics just to read the value of the force, or just to detect the excessive force, to have something like an alarm. So it's under development right now.
Thank you.
Thank you.
Okay.
Thank you very much.
The last presentation in the session will be presented Professor Katarzyna Arkusz from Zielona Góra.
Thank you. In the beginning, I would like to say thank you to the organizer for such a wonderful conference and for inviting me. So today, I'm going to show you the preliminary result of new nanomaterial or nanocoating methods for titanium. Okay, thank you. So, generally when we talk about the surface or implant surface modification, we can divide it into two mechanisms. The first one is micro-modification, and the standard technique like sandblasted or acid etching is commonly new, is very well known, and it gives us the possibility to use this technique to improve the mechanical stability or mechanical scaffold or implant, and additionally it also improves the connect between cell and between materials. But when we talk about nanoscale, the most advantages of this nanomodification is that nanomaterial can promote the osteointegration, or can promote the cell, can enhance the cell promotion, sorry. So when we talk about the nanotechnology, commonly known nanostructure are the circular titanium dioxide nanotubes. And this nanostructure exhibits all of the advantages known for nanomaterial. So they have good biocompatibility, antibacterial properties, and few reports reported the brightness of this layer. So it's definitely, but maybe not definitely, because these research are not finalized yet, so we have to check it, but it is the big problem in using nanotechnology in implantology. So when we have this kind of problem, we decided, or we were wondering what to do to avoid the brightness of this structure, and we were inspired by honeycomb structure. So we asked ourselves if it is possible to form not circular, like you can see in this picture, but the hexagonal titanium dioxide nanotubes, and that, as you can see, it is possible. But I'm going to show you the way to obtain this nanostructure, and I'm going to show you the main properties of this nanostructure or nanomodification. So generally, our research were focused on synthesis on hexagonal titanium dioxide nanotubes, or HTNTs. I will use this abbreviation, using sono-electrochemical anodization. Then we had to carry out the thermal modification. After that, we analyzed the properties. In this presentation, I will focus only on corrosion properties, on mechanical properties and biological properties. However, we carried out the research in extended area. And the last one, or the last stage of our research was the implantation into pig's mandible. So, when we get this idea to obtain hexagonal titanium dioxide nanotubes, we were looking in the literature if somebody done it before us. We find a lot, maybe not a lot, maybe not maybe not a lot, but we find few paper where the hexagonal titanium nanotubes were described as a circular nanotubes, but in hexagonal arrangement. And sorry, and only a few, to be exactly 5 paper, show the possibility to form hexagonal titanium nanotubes. And as you can see, the most uniform nanostructure were obtained using atomic layer deposition method with alumina template. So previously we worked with circular titanium nanotubes. So we were wondering how to change this shape from circular to hexagonal. And we decided in the first step, because during the Lebedeva theory, the hexagonal titanium nanotubes are the precursor to circular nanotubes. So we decided to make this process, to make the anodization process faster. So we add the ultrasound to our method, but it doesn't allow us to obtain this structure. So we were focused on the spaces between single nanotubes, and our hypothesis was that when we make this space so close, it will allow us to obtain the hexagonal titanium nanotubes. So we used the chelating agent, and finally we obtained the hexagonal titanium nanotubes. In this SIM picture, you can see the bottom view of these nanotubes. In this picture, there's a cross-sectional of hexagonal titanium nanotubes, and here you can see the top view. So generally, the mechanism of formation is very simple. Now I can say it very simple. So it consists of dissolution of titanium using fluoride ion, and the chelating agent, EDTA, increases the dissolution process, resulting in disappearing the spaces between nanotubes. So, what are the properties of this material? Generally we wanted to check the full abroad of properties, and we wanted to check if the diameter or diagonal, it depends which kind of morphological parameter we will use. We wanted to check also the correlation between morphology, geometry, and the properties. So the first step was to optimization the process, and few conclusions were generally known to us. So the diameter depends on voltage of voltage during the anodizing process, and the length of this layer depends on the time. But when we use the high voltage anodization, this process were completely changed. Another key aspect were the concentration of fluoride ions and the concentration or ethylene glycol used in this process, used in electrolyte. So as you can see in this picture, there's a very small, there's a very small range of this concentration, which allows us to form hexagonal titanium dioxide nanotubes. So when we optimize the process, we fabricate the HTNT with a diameter from 30 to 100 nanometer. So we wanted to examine this nanostructure in a whole range of nanoscale. And we produce the titanium dioxide nanotubes with the length from 3 micrometer to 4 micrometer. This length is dedicated to surface modification of implant, so we use this parameter. As a additionally, we compared this nanostructure to commonly known compact layer. According to sonoelectrochemical process, and it was the first parameter or the first observation which aware of was the fluoride concentration. To dissolve titanium, we have to use a very high concentration of fluoride, and if we want to use this material in human application on your human body, we cannot leave this concentration into the material. So, firstly, we examined the corrosion properties. We weren't satisfied that hexagonal titanium dioxide nanotubes revealed higher corrosion rate than compact titanium dioxide, but generally these values are in range of corrosion resistance, which are dedicated to implant. Never mind if we talk about dental or orthopedic implant. Then we carried out the non-indentation test and scratch test to check the mechanical stability of this nanostructure and to confirm if our idea was correct. So, as you can see, the hexagonal titanium dioxide nanotubes reveal Young modulus very close to Young modulus of bone mineral. This nanomodification also decreased the hardness. Never mind if we talk about Martens or Vickers hardness. And what is the most important, maybe not the most, but what is also important, hexagonal titanium nanotubes revealed higher plasticity when we compare it to compact TiO2. Scratch test, generally the hexagonal titanium dioxide nanotubes with a very small diameter, we examined 13 nanometer, revealed higher critical load compared to titanium dioxide. What does it mean? It means that this layer is more stable and is very solid onto the implant surface, and the critical load decreases with increasing the diagonal of HTICs. So, at this stage, we were wondering which diameter or which diagonal of titanium dioxide nanotubes is preferable for implant application. I haven't shown you the result of wettability measurement, but generally all of this structure, never mind if we talk about the very small or very high diameter, revealed super wettability. The drop put onto the surface immediately disappeared. So we decided to use in our future research the diagonal with the HTIC with diagonals of 50 nanometer due to this parameter. I told you that we had to use thermal modification process. Why? There's few reasons. Firstly, we want to carry out the transformation from amorphous to crystalline surface, and additionally, we want to avoid fluoride or make the fluoride disappear in our nanostructure. So, only main conclusion. Generally, after annealing in inert atmosphere in argon for 3 hours at the temperature 350 and 450, using XCRD and using the Raman spectroscopy, we obtain the concentration of Aneta space. Generally, Aneta space is dedicated to or preferable to implant application. And when we use the higher temperature, as we expected, we obtained a mixture of Anetas and rutile. How does it influence onto corrosion resistance? Thermal modification improved the corrosion resistance, so it is also in the range dedicated to or required to implant material. What about mechanical properties? Depend on thermal modifications, so generally depend on Aneta's content in our sample. The Young modulus were significantly lower, and when we use the higher temperature and when we obtain the mixture of Aneta's and rutile, the Young modulus is almost at the same level. The hardness of this material increases, so it is our observation. And when we talk about the critical load, it also increases. So, based on this result, we choose the temperature 550 to next step of our research. And the next step was the biocompatibility test using a simple MTT test and the implantation into the mandible of pigs. So we sterilized our sample. This sample were 50 nanometer. The diameter of HTNT were 50 nanometer. The length was 3.5 micrometer. We annealed it in argon atmosphere for 3 hours at 550 degrees of Celsius. Then we see the 50,000 mesenchymal stem cell, and we carried out the measurement from 3 to 5 weeks. So when we got the result, we were positively surprised because the viability of stem cell onto this surface were very good, and thermal modification improved this parameter, and also the proliferation or the number of cells were increasing, and it was also very satisfied result. So, in the last one, we wanted to check if the mechanical properties of this nanolayer. I said in the beginning that the problem of circular nanotubes were cracking this material during implantation. So we carried out the simple implantation test, and we observed during scanning microscope, scanning electron microscope, the surface of this implant. And generally, we didn't see any damage and cracking of this layer. Sorry. But we decided also to carry out the electrochemical impedance spectroscopy test, and we saw some difference in electrical properties of this layer. So this is our goal for the future, to check if this material is very stable or there is some micro cracks in this layer. So to conclude, we form the new implant nanomodification, hexagonal titanium dioxide nanotubes. We optimize this process to obtain the HTNT with morphological as we want. We carried out the thermal modification of this process, and we also carried out the sample test, confirming its possible use in implantology. Of course, I haven't done this research by myself, so I'd like to thank part of biomedical engineering group and our collaborator who was Technical University of Košice, Silesian University of Technology, and Poznań University of Technology. Thank you for your attention.
Thank you for your presentation. Very interesting results. Are there any questions?
I know, it's lunch minute.
There is no need. Thank you so much. Yes, I have a question. Thank you very much because thank you for the presentation. It was really spectacular, especially when we just compared these two, all of these results. It's horribly hard to find a conclusion, and you did it. So congratulations. By the way, I've got a question related to surface modification with the use of a heat treatment. Was it designed in order to change the, let's say, to perform the traditional annealing in order to change the structure, or rather it was designed in order to modify the surface in order to obtain a surface layer on that one?
No. Firstly, we fabricate this layer onto the titanium foil, and then we carried out the annealing process. The annealing process doesn't break down the morphology of this nano layer, and it all, as I told, we obtained this crystallization of this process.
It was
Yes.
designed as a change of a structure, internal structure.
Yes, general
and
structure.
Later on, the consequence for the change was the increase of corrosion resistance and the change of morphological parameters.
Yeah, I asked us this. Okay, generally in nanotechnology we can obtain, for example, circular titanium dioxide nanotubes using annealing process. So this is another method. But in our application, we only use thermal modification or annealing just for changing the crystalloids of this nanostructure.
So the change of internal structure. Thank you. Thank you very much.
Thank you.
Thank you. Fantastic. We are on time. Many thanks to all speakers for their presentation. Many thanks to all participants, and I invite to lunch. Thank you so much.